Ekis, ekis. Ekis, ekis. Coming at you with 50,000 megawatts of power. It's SCSC 681 seminar. The greatest show on earth, at least so I've been told. Coming at you today, we've got three, count them, three, not two, not four, but three student presenters giving us the best of their research. You're in for a great show today. So I think maybe I hear Steve Haig is going to introduce our first speaker. Is that correct, Dr. Haig?
You are correct, sir.
Fire away.
Okay. Yeah, I'd like to introduce Jake Cottrell. He is a distance master's student in our distance plant breeding program. He thinks he's going to graduate this summer. I think he will. He's been working on his master's in plant breeding since 2018. So he's right on track with that program. He is currently in Lubbock, Texas. He's working as a research associate with Corteva, and he's worked there for over 5 years now. His primary responsibilities in that position are managing yield trials and specifically with drought stress, and then managing their UAS program for that research station. He also wanted me to tell everybody that he is married and he has a son and a daughter. And if you've ever met his wife, that was probably one of his greatest accomplishments. So he definitely outkicked his coverage. So with that, I'll turn it over to you, Jake.
Thank you, Dr. Haig. Okay, can everyone see my screen?
We can see it.
Okay, so as Dr. Haig said, I've been working at Corteva AgriScience and Drought and Maize for the last 5 years, and decided to do my plant breeding master's degree in 2018, and wanted to, my research is in the effects of drought on silk emergence and yield in maize in the Texas High Plains. The research objectives were to confirm yield performance of drought-tolerant versus drought-susceptible maize hybrids in a managed stress environment and its correlation to the silk emergence trait. I also wanted to compare yield and silks per ear of each drought-tolerant and drought-susceptible hybrid pair. They're paired by relative maturity in corn. And then confirm the yield advantage of drought-tolerant hybrids when drought stress occurs around the anthesis time period. So again, my hypothesis for this research was when drought occurs in the Texas High Plains around flowering, the drought-tolerant hybrids will yield higher, exert more silks, and produce more kernels per ear compared to those of the drought-susceptible hybrids. This research was done in the summer of 2018-2019 at the Corteva Research Center in Plainview, Texas. There are two environments, a fully irrigated and a drought stress environment, and each environment had 6 reps with 10 entries. The traits that were collected were yield, silks per ear, kernels per ear, grain weight, and 100 kernel weight. So a little bit more about the research farm in Plainview. It was installed in 2012 and 2013. Roughly 114 irrigated / cultivated acres available for plots. It has subsurface drip tape arranged in 8 quads, and in each quad has 7 2-acre drip zones. The components of this irrigation system are custom pressure compensated tape, 12-inch emitter spacing, and an EcoDrip EC3 controller. The reason why we talk about these components is it allows for a very precise watering system. We're able to deliver accurately as little as 1/10 of an inch. Also, it's a very uniform location. The soil is Olton clay loam, and one of its major advantages is its reproducibility over years. And if you look to the right, that is an image that I took with a drone. That is the entire research farm in 2020. And as you can see, mostly under maize and a little bit in the bottom right-hand corner is sorghum. Okay, so again, I spoke briefly about my environments. Just more detail between the fully irrigated and the drought stress environment. The fully irrigated was irrigated at 100% ET. If you look down there, the total water put out in 2018 and 2019 is 126 millimeters and 843 millimeters respectively. And the major difference in the environments in the drought stress environment, we went with a managed stress environment compared to an ET-based environment. Basically the difference between a managed stress environment is that it was treated as a fully irrigated location before and after anthesis, and the only amount of water that was given was to prevent death of the crop. And as you can see down there at the bottom, the totals for 2018 and 2019 were basically the same at 725 millimeters and 729 millimeters each. And if you look, just a quick, easy comparison between 2018 and the fully irrigated and the drought stress, we had roughly about a 200 millimeter difference. That's about a difference of 8 acre inches. And just a quick note that rainfall was only taken from January to August. The experimental design for this research was nested, and then it was randomized within the nest. And as I stated before, 2 locations, fully irrigated drought stress, 6 reps at each location with 10 entries per rep. In those 10 entries, had 5 drought-tolerant hybrids and 5 drought-susceptible hybrids, and the nest made up one of each, a tolerant and a susceptible. And again, they were paired by CRM, relative maturity. And we mapped the reps vertical through field, so they were randomized, but we were pairing a susceptible and tolerant right on top of each other for the ability to make a direct comparison. These were 4-row plots, 4.4 meters long, 76 centimeter row spacing, and around 79,000 plants per hectare. Yield captured on the inner 2 rows, and the outer 2 rows were utilized for our silk cutting samples. Either one or the other row was utilized on the outer row. These samples required us to collect 5 sequential plants with no gaps or no double stand, and they would be 5 plants in from each end of the plot. So a little bit on how we took these silk cutting samples. We utilized a silk cutting device developed by Corteva AgriScience. We collected these samples 3 days after 50% silk. We collected them early in the morning before the silk sluice trigger. The way this device works is you grab the bundle of silks and straighten it out and put it in that little loop in the silver part of the cutter, and then you would close the cutter, and the sample would squeeze it. It would cut with 2 blades with a 2 millimeter sample, and it would squeeze to that hole. And the way it's designed is you put a 20 milliliter scintillation vial in that hole, and you're able to collect your sample fairly rapidly and very easily. It also allows for storage up to 1 year if immediately stored in alcohol in a refrigerated environment. Oop, I went the wrong wrong direction, apologize. So once we collected the silks, we had to count them, and doing that required us to take each sample, put them in a Petri dish, and take a image of them by digital camera in a special photo box. And on the left there is an unprocessed image of our silks, and then on the right is a processed image. And what we did was we ran it through an algorithm that essentially put a count on every single silk. So all the pink that you see in the dish is essentially either a single count or multiple counts if there were silks right next to each other. And again, the total count was put in the top left corner of each image. At the end of running the algorithm, it was all compiled into a spreadsheet, so I didn't have to go look through every single image for a count. To verify accuracy of this, we used 15 calibration sets where we manually printed out the unprocessed image and manually counted each silk per image, and then ran the algorithm and compared the counts, and the R-squared of the algorithm was at 0.97, which we feel is much more accurate than doing everything by hand. And also another advantage of this method is it's very high throughput. So now I'm getting into the data for my research. So this table is set up in the middle column. Just a second. The middle column right here is the silks per ear, and then on the far left is the yield. And this is a comparison of the environments. So we have the full irrigated environment on top and the drought stress environment below it, and the associated statistical values below that. Right off the bat, if we're looking at yield first, we see that over both years, we had just over 13 megagrams per hectare, and in the full irrigation, and in the drought stress location, we had 8.6 roughly megagrams per hectare, which is very significant and of a separation, and basically shows that we did achieve a significant amount of stress. To put that in terms of bushels per acre, this is roughly 200, and this right here is roughly about 120. And you can see the heavy percent reduction between one to the other in yield. And if we look over here at silks per ear, we see again another significant reduction, showing that yes, there was stress during the flowering period. So this table is set up in the same way, column-wise, as the previous one, with silks per ear and then yield, but instead of just a straight environmental comparison, these are comparisons of the tolerant hybrids and the susceptible hybrids in each environment. So on top here is the full irrigated, and then with the associated statistical values, and then below that is the drought stress environment, both tolerant and susceptibles, with the statistical values below that. If we look here, not any separation between silks per ear in the full irrigated, which is what we wanted to see. We do have a little bit of separation, but not highly significant between the yields. But if we come down to our drought stress location, which is this is the important part of the research. We've got a very significant separation between the drought tolerant yield at just over 9, and then the susceptibles are just at 8, which shows that we were able to separate, using our managed stress environment, we were able to separate the yield out very cleanly. And we also were able to show a very good amount of separation between silk numbers here for the tolerance up top 460 to the combined susceptibles at 426. And one thing to note down here at the bottom is we did some reductions of the fully irrigated to the drought stress for the tolerance, which we reduced this yield from 13 to 9, which is roughly a 31% reduction. And here we reduced from 12.8 to 8, which is a 37% reduction. This 6% reduction, or difference in reduction, I should say, is essentially translates to money in the grower's pocket. It's basically, it could mean a difference in a limited environment and making a little bit of a profit and barely breaking even or not even breaking even. So this table, in 2019, we took yield component data in kernel number, grain weight, and 100 kernel weight. And these are the results. We also included silks per ear and yield in this as well as comparison. And as you can see in every column, we were able to, sorry, I need to set up a little bit better. But this is this on the top is the fully irrigated environment. And as you can see in every case, we significantly separated from the fully irrigated to the drought stress environment. The next two groupings of data below that is tolerant and susceptible comparisons in this is the full irrigation, and then the tolerant and susceptible comparison in the drought stress location. And as we can see, in the fully irrigated environment, not a lot of separation, a little bit of significant separation, but not as high of significance as there is in the drought stress environment for all three yield component pieces. So this table is just one of the individual hybrid comparisons that I had. As I spoke, I had 5 pairs, and this is one of those maturity pairs data. And again, it was set up similar to a table in a previous slide where we have the fully irrigated environment on top, the comparison between drought and susceptible, and then below that we had the drought stress environment comparison of the tolerant and susceptible. And again, we see very similar results. This one actually in the full irrigation environment did have a little bit better time for the tolerant than it did the susceptible, but we really see a very nice reduction in yield right here between the tolerant and susceptible in the drought stress environment, and also a good amount of significance in silk numbers per ear reduction here in the drought stress environment. And we don't see that here in the fully irrigated. There's a little bit of separation. In fact, the funny thing is it actually produced more silks for the susceptible in the fully irrigated environment, but yielded less. And this could be due to a number of factors. Most likely, it would be tip kernel abortion later in the season. And again, down here, another important separation of about 5%. Again, putting, that's a lot of money in the grower's pocket. Again, a profitable year versus a maybe breaking even year. So this is two linear regression models that I ran. The one on the left here is x-axis is emerged silks over yield, and as we can see, the R squared is roughly about 0.20, basically meaning that exerted silks, or the silk exertion trait, in these environments roughly explains about 20% of the variation of yield. And if we look over to the right, we have a much higher correlation, but this is kernel number over yield, and we see 0.79 as the R squared for that, and which we do know that kernel number has a very high correlation with yield. Okay, so conclusions. Both yield and silks per ear were reduced in drought stress environments compared to the full irrigated. The drought tolerant hybrids displayed superior reproductive resilience under drought stress at flowering when compared to the susceptibles. Improved silk emergence of drought-tolerant hybrids contributed to their improved yield under flowering stress, but other factors, as I spoke earlier, other factors such as reduced kernel abortion and better canopy photosynthesis could have contributed to a better performance by the tolerance. I'd like to acknowledge Corteva AgriScience. They provided all the resources for this research. My distance advisor, Dr. Jeffrey Schuessler, my committee chair, Dr. Steve Haig, Scott Adair, who is the trial site manager for the farm that I utilized, Cho Hugh, who worked countless hours with me on the algorithm fine-tuning, and then our interns for both those years, Caitlin Lakey and Jacob Ford, who helped me in collecting a large amount of silk samples in the early morning hours. Okay, questions?
Good job, Jay. Great seminar. Questions? Don't be shy.
All right, I have a quick question.
Go.
Yeah.
Can you please go back to the slide where you showed how you process the imagery to count the silks?
Yeah.
I might have missed it, so would you mind elaborating a little bit on that?
All right, so what slide again? I was having computer issues.
Like how you analyze those images to count the silk? Yeah, yep.
On this, yeah. So yeah, it was an algorithm developed within Corteva, and again, we took a picture of the petri dish on the left. Is essentially each sample that we did, and took those images and ran them through an algorithm in Python, which essentially assigned a count to every single dish. It looked for the area in blue as opposed to the area in green to collect those counts on. Okay.
You still had to count manually those clusters, right? I can see the clusters there. Some of the clusters.
Yeah. So in some of these clusters, the counts are very accurate in the clusters. When I took the images of this, I tried to make sure that the silks were separated so they weren't bunched up, so we wouldn't get just a big red blob in the middle of the processed image, and that would lead to some inaccuracy. But if you kept the clusters small, you still got a very highly accurate count.
Oh, okay. Thank you. Thank you so much.
Mm-hmm.
More questions?
Hi, Jake. Wayne Smith. Great job. Enjoyed your presentation.
Thank you.
Can you, did you have as a part of your study whether or not there was any genetic relationship between the hybrids that were susceptible and the hybrids that were tolerant? Are they the same backgrounds? Or do they come from various sources?
We had most of them are various sources. We mainly focused on maturity. However, we did have one experimental pair in this research that essentially was a wild type and a traded or an event. So.
Other questions? What about the tassels, Jake? Do you think they contribute to the yield issues?
So ASI, which is the anthesis silking interval, we know from previous studies in the past is essentially extended under drought. So pollen shed, while can be affected by the tassels, just what I like to call sneaking or staying in the leaves and already starting to dehisce. Really what happens under drought is the pollen still sheds when it's going to shed, but we have a delayed silking reaction, which causes silks to miss the pollen window.
Mm-hmm. Okay. So is Corteva coming up with something to spray on the plants to increase the yield in the drought?
Not that I'm aware of.
Well, they should. They really should.
Water.
Yeah. Very good. All right. Any further questions? Nice presentation, Jake. Thanks.
Thank you.